Okay, let's get started. Hello, everyone. I'm Mrna from the SingHealth Duke-NUS Global Health Institute. And on behalf of the Institute and the co-organizers, the American Chamber of Commerce in Singapore and the Swee Hock School of Public Health, I'm delighted to welcome all of you. Thank you for sharing your time with us. In today's context of uncertainty and overwhelming information from multiple sources, we hope to be able to pass out the most relevant information for you today. This is the second webinar in our four-part webinar series on the science and business of COVID-19. This webinar looks at vaccines and treatment options and will be moderated by Professor John Lim, who is the Director of the Center for Regulatory Excellence at Duke-NUS and Policy Lead for the SingHealth Duke-NUS Global Health Institute. John will now introduce the panelists and the topics of today. Over to you, John.
Thank you very much, Mrna, and welcome to all of you who are joining us. We've had well over 1,000 people registered for this webinar, and I still see a lot of people coming into the chat room at the waiting room at the moment. So our topic today is on the issue of preventing and treating COVID-19, and this big question: when will we get a vaccine? Are there any effective treatments? And we're delighted to have four very eminent and experienced speakers to join us on the panel this afternoon. What I will do is I'll introduce all of them first, and then I will ask them to, in a just consecutive manner, share their thoughts on the issues, and then we will take your questions. We've had a lot of questions that have come in already, but if you look at the Q&A icon at the bottom of your screen, we will be able to take further questions in the course of the presentations, and we will try and address those if they've not already been covered either in the presentations or in the questions we've received. So we're delighted today to have with us Professor Liu Yi-Sin. Yi-Sin is Executive Director of the National Center for Infectious Diseases in Singapore, and she's been at the forefront of this whole battle that we've been fighting against COVID-19. And of course, her experience goes back even to SARS in 2003. So Yi-Sin is extremely experienced in this area. She's also an adjunct professor at the Swee Hock School of Public Health at NUS. Our second speaker today is Assistant Professor Ashley St. John. And Ashley is with the SingHealth Duke-NUS Global Health Institute, as well as the Emerging Infectious Diseases Program at the Duke-NUS Medical School. Our third speaker is Professor Eng Yong. Eng Yong is also from Duke-NUS, from the SingHealth Duke-NUS Global Health Institute, and he is Deputy Director in the Program in Emerging Infectious Diseases at Duke-NUS. And we're delighted that we have a fourth speaker who's been able to join us this afternoon, Mr. Amgad Gamil, and he is the Senior Director for Medical and Scientific Affairs for Emerging Markets Vaccines from Pfizer. So they will be covering a range of topics. I've asked Yi-Sin to give us a brief update first on the clinical experience that she's had in Singapore from the NCID, and touch a bit on issues of therapeutics, what is available at this point in time. Ashley will go a bit further into this area and begin talking about vaccine development, followed by Eng Yong, who will take this further. He will give an update on what's going on in Singapore and internationally. And then Amgad will come in to talk about the industry perspective of vaccine development, specifically with respect to COVID-19. And so without any further ado, I would like to hand who will start off the session, and she will then hand over to Ashley, Eng Yong, and Amgad. So without further ado, can I invite Yi-Sin to share her thoughts on this topic? Yi-Sin?
Good afternoon. Thanks, John, for the very generous introductions, and thanks Duke-NUS and Swee Hock School of Public Health for giving me this opportunity to say a few words. I would first describe the current local situations and perhaps then throw open some of the challenges and tasks ahead in relations to therapeutic development as well as vaccine development. I'm sure most of the audience here are quite familiar with the local situations. The first case was detected on the 23rd of January this year, and a circuit breaker was introduced on the 7th of April. So precisely today is the 14th day of our circuit breaker. And obviously many of us are looking towards having good news ahead in terms of how the public health interventions would have helped us this far in trying to flatten our curve. So just to quickly mention to you the local situations as of yesterday, we have more than 6,000 cases being detected and reported. And majority of the cases in the past few days predominantly contributed by our local foreign workers in Singapore. About 95% of them were due to the transmissions among the foreign workers, and about 5% or so of the local and permanent residents. Import cases, in fact, is very, very small, hardly seeing any import cases currently because of the travel restrictions. And out of the 6,000 over cases, currently we have 11 reported fatal cases. So overall, the case fatality rate is pretty small in Singapore comparatively, but not to be too happy and complacent at this point in time, largely because we are detecting many early cases as well as much younger cases in our cohort currently. And I would say that out of the 11 deaths that we have so far, only 6 of them went into ICU, and the other 5 cases were elderly individuals who had prior determined to have no advanced care provided. So all in all, I would say that of the 90 over cases that ever require ICU, we have a majority of them recovered from the supportive care through ICU. So in actual fact, just looking at the standard of care alone in Singapore, including the care provided in the ICU setting, we have achieved reasonably good outcome. And that also gives challenge in terms of the therapeutic interventions and therapeutic approach. Now coming to how current recommendations on the therapeutic approach and medication use, I would just want to first mention and stress on the point that there is no proven effectiveness in any of the antivirus so far. A lot of focuses are currently being put on remdesivir. Unfortunately, I think at this point in time, there are quite a fair bit of mixed signals coming out from Gilead with regards to the Gilead's trial, and there is no control arms in the Gilead trial so far. So it makes the comparisons and the interpretations of the results very difficult. So this is one area that we have to really focus, whether or not remdesivir trial in the NIH will give us a better answer in terms of the proper RCT trial. If without any anti-retroviral at this point in time, what other things can we look at? Can we now look into immune modulator? There are a lot of studies in this aspect, and there are also quite a number of studies globally looking at the use of steroid, whether it's high dose or low dose steroids. Now, I just want to say that I mentioned earlier on, just providing good supporting care and good ICU care, we have reasonably good outcome. And therefore, it becomes very difficult and challenging for the clinicians who now would want to go into a therapeutic treatment that is of no proven clinical efficacy. And we know that more than 80% of our cases, the conditions is mild to moderate and self-limiting, and only a small proportion of them would require oxygens, supplementary oxygens, as well as ICU care. And the other big challenge in terms of therapeutic approach is that for any of the medical interventions that is against the virus, obviously we need to go in to treat the patient as early as possible. However, at the early stage of the clinical illness, it's extremely difficult to select out patients that will subsequently progress to more severe disease. So those are the clinical challenges at this point in time, how we look into therapeutic interventions. I think all of us are waiting for the possibility of available of vaccines one of these days. And I think it's important for us to hear from the other panelists in terms of the private-public partnership in the ability to deliver the vaccine as quickly as possible and effective vaccines that we don't see too much with antibodies confusions or antibodies enhancement activities. And as of now, I believe there is no human coronavirus vaccines, except for the veterinary may use some of the coronavirus vaccine. We don't have a lot of experience at this point in time. I'd like to hear from the rest of the panelists, but I'll just say that without good antivirus, without the availability of vaccine, we have to use many other non-therapeutic, non-pharmaceutical prevention methods to continue to bring down the disease transmissions and to give the best supportive care to our patients. Over to you, John.
Thank you very much, Yi-Sin. We will come back to you later in the question and answer. There was a question also, some questions have been asked about how do you manage asymptomatic patients, so please consider that later. And several questions have come about hydroxychloroquine, but I think you've basically said there is nothing proven and effective at the moment, but maybe you can address that later. And there's some questions about alternative therapies as well. So we'll come back to you later, Yi-Sin, but now I'd like to hand the time over to Ashley to talk more about therapeutics and vaccine development. So Ashley, over to you, please.
Thank you, John, and thank you to the organizers for inviting me to join you today. So I want to kind of go back to basics and talk about what is a vaccine. So ideally, a vaccine, you'll give the immune system a glimpse of a pathogen, the smallest amount of a pathogen or any external antigens possible to allow the immune system to recognize it as foreign material and develop a memory response. So there are two aims of vaccination. First, we want personal protection. We want to generate immune memory in an individual. On the other hand, we also want herd immunity. So vaccines are really intended to protect the community, and they don't usually lead to 100% protection of every single person in the community. And so most good vaccines are around 60 to 80% protective. And so that's what we will be hoping for in vaccine candidates that come out against SARS-CoV-2. And so then the question is, what kind of specific protection is needed against SARS viruses? And some of this information we're beginning to learn from the earliest studies that are coming out, and some of it we know from related viruses like SARS or MERS. And we have an idea that both branches of the immune system are probably important, memory immune system, I should say, so both antibody responses and T-cell responses. But there's some preprints out that show that perhaps antibody responses are a little bit borderline. So some people who had very high antibody responses, at least for a hospitalized cohort, were more likely to have severe disease. So we don't know what that means. We don't know yet if that is because the antibodies are causing a problem, or if it's because with a more severe disease, they're able to generate a stronger immune response. So these are some of the questions that will be important to answer as we go forward into testing and using a vaccine against SARS-CoV-2. So what are some of the unique challenges for this context? So first, we need a vaccine very fast in this situation. And for that reason, I think most of the candidates are leaning towards things that we know will be very safe. And I'll speak about that in a minute. And the other aspect is that we need a vaccine that's also protective in the elderly. A lot of times our vaccine development programs are designed for developing childhood vaccines. So this is a little bit unique situation. The elderly can be refractory to vaccines. They can be a little harder to immunize and get a strong immune response. And of course, we have to be sure that it's a very safe vaccine. So there are examples of how this has been solved for other viruses. So for example, the flu vaccine, we have a live attenuated vaccine that's given to younger populations. And then we have a subunit vaccine, which is just a protein antigen that is given to the elderly. It may not be quite as effective, but it's something that is a little safer in that particular population. So this may be an option as well, and it's a potential for expecting that perhaps even multiple vaccine candidates could serve different populations in the long run. In terms of what kinds of actual vaccine candidates are being considered here for SARS-CoV-2, there are, as I said, many candidates that are leaning towards trying to make things safe. So live attenuated vaccines are not really the prime targets here. People are looking into virus-like particles, which are particles that really replicate the whole structure of the virus, from the nucleic acid to the proteins on the surface of the virus. There are nucleic acid-based, such as DNA and RNA vaccines, and there are also subunit vaccines, which, as I said, are proteins that usually need another substance with them, like an adjuvant, to trigger the immune response and really indicate that this is foreign material. So it'll be interesting to see which of those are most effective in trials, because to date, I'm not aware of any preclinical trials showing efficacy yet. So we'll be looking for that to see what is pushed forward. And I also want to mention that vaccine optimization also involves optimizing the dosage of the vaccine and the schedule of the vaccine to achieve durable protection. And so those are things that in the long run studies will be needed as well to determine what is effective for each individual vaccine candidate. So, and I also want to mention that having a vaccine candidate or having a vaccine that is given to the community also raises some other concerns. So for example, then we may eliminate the ability to use serology or antibody responses in the blood to test for exposure. So in the cases there are breakthrough cases, it may make it a little more difficult to understand the roots of infection in the tracing. So we do need to keep that in mind. Of course, there are always other safety considerations. So one consideration is, as I said, this role for antibodies. There's a concern whether antibodies could potentially generate something like a antibody-dependent enhancement of infection or enhancement of pathology type response as occurs for dengue. This isn't necessarily going to happen. It has been shown in some experimental systems, but it's just an important thing that we need to test for with any vaccine candidates before we really introduce them into humans. And then I want to take it back to testing and mention one final point on that. So most of the assessments about returning to normal after the shutdown period that has occurred almost everywhere in the world emphasize the importance of testing in the absence of an effective vaccine to determine who can re-enter the workforce, which healthcare workers are protected, and also facilitate contact tracing and isolation of these clusters. So I want to emphasize that testing of serology is different from testing for acute infection. So when we test for acute infection, we're testing for the nucleic acid usually in a PCR-based method for the virus itself. And then someone is considered positive if they have the virus. But alternatively, we can use serology to confirm cases if we miss this window when they're infectious. And that involves testing for antibodies in the serum. A recent study showed that about 50% of people are positive at 1 week after infection, and after, I should say, onset of symptoms, and about 100% have seroconverted by 2 weeks. So this can inform contact tracing, and it can also reveal who's likely been infected and also protected from infection. So with that, I will give it back to John. Thank you.
Thank you very much, Ashley. So in a sense, you're doing a tag team with Eng Yong, who's also talking about vaccine development. So let's hear what Eng Yong has to say, and then I'll look at the various questions we have to pose to both of you subsequently. So Eng Yong, can I hand the time over to you?
Thanks, John, and thanks, Ashley, for doing bulk of the work. So I'll take it on from where Ashley left off in terms of, she's already described the science and the kind of the different ways of developing vaccines. And I'll take it on from there and ask the question, well, how do we bring it forward into the clinics? How long that would take? The third question I hope to tackle is, why are there so many different efforts to develop a vaccine, and is that necessarily a good thing? Are we just trying to race with each other and see who wins? So let's just take question one first. How do we bring a vaccine into the clinics? Well, first of all, the two things we need to show. One is that it works. Two is that it's safe. Now, typically, the entire process takes 15 years average, right? I think the shortest is measles, about 8 years. And we're trying to shrink a process that takes about 10 years or more down to, less than a year or slightly over a year. That's a tall order, without doubt. So then the question is, how do you do it? So I think in any aspect of public health, it's healthy to think of it as risk-benefit, right? So the benefit of a vaccine is in the context of this pandemic. Obviously it would be great. And we can come back to the question of, is 60% efficacy enough? But nonetheless, it's a question of risk-benefit. So if you have a vaccine today, we can, one, prevent a lot of infection. Two, we don't have to have to shut down, and probably life can go back to normal. So under such circumstances, you would expect that you can probably have some appetite for some risk. Now, I'm not saying that we're going to accept a vaccine that's not safe, but you're going to be willing to accept a vaccine based on its theoretical potential and what we know of its safety profile from use previously, and infer it across rather than having to do the experiments. So in that sense, you've seen that, you know, the first vaccine to hit clinical trial did it in 42 days. That's remarkable, right? From the point where they said they would design a vaccine to the point where they actually entered human trial in 42 days. It's unheard of. But it speaks of the urgency of the matter, and the fact that too, we also know a lot more about vaccine science today compared to the past. And so a lot of the things that we do today to bring vaccines into the clinical space is based on studies that were designed in the 1950s and 1960s. We are much better than that. We understand a lot more about, you know, what constitutes vaccine safety, what makes vaccines work much better now. And so one way in which we're going to accelerate vaccine translation, therefore, is to bring in these new molecular tools to assess vaccines in a way that was never done in the past, right? Because we know that everything is controlled by genes. Whether you get a headache after you receive a vaccine, whether you get a bit of body ache, whether you have fever, they're all controlled by genes. How well vaccines work, what kind of antibody response, what kind of T-cell response, again, they're all controlled by genes. And these genes are turned on and off in the first 5 days or so after you receive a vaccine. Whereas whether a vaccine would work or not, it will take years for us to figure out. So then by looking at what genes are turned on and off after you receive a vaccine, we can very quickly know whether a vaccine will have a high likelihood of working and whether it's going to be safe in terms of the side effects, right? So that's how we're going to accelerate the vaccine. And, you know, our hope is that more and more vaccines will enter human trials, and then by hopefully middle of next year, we'll start to know which ones will work, has the potential to work. So that in a nutshell is how we're going to translate vaccines. And the same thing will apply for therapeutics as well. Again, it's a risk-benefit analysis, it's a consideration. Like again, you know, if we have drugs that we know have a very good safety record, then even if this is a new version, then its chance of getting into human trials are much faster. And the best example of that are antibodies. These are human antibodies. So instead of us generating the antibody, we make them in the lab, or we make them in production facilities, so that we have a lot of doses of this one single antibody. And we know that if it's a fully human antibody, and it has all the germline characteristics of a human antibody, then the number of side effects and all that will be very, very low, and we can accelerate these into clinical trials. So the immune-based therapeutics and the vaccines will then operate in this realm that we fall back on what we know about safety from what's been used previously. So finally, you know, right now there are about 70 vaccine efforts going on in the world, another 30 or 40 coming on the scene. So about 70 have progressed to animal or stage or even later. And then another 40 or so are beginning. So you're talking about hundreds of different efforts to develop a vaccine. And the same idea about drugs. There are also perhaps even more different efforts to find a drug and vaccine. The question then is that, are these really needed? Are we just, you know, in an ego race to see who beats whom? I think my take of this is not. It is not an ego race. I think to win at this, we need to take multiple shots at goal. See, we have only about 5 months to know this virus, so there's a lot that we don't know. So there is no basis to say that one vaccine or one drug is going to work better than the other one, because it's really, your guess is as good as mine. There's not enough data for us to know which vaccine would work best, and Ashley already outlined the different ways a vaccine could be made. Then the question is, which of these vaccines will give the immune system a good enough snapshot so that they will recognize the virus the next time it sees it? And the answer is we don't know which one would work in that way. So therefore, it makes sense to me that we take multiple shots at goal so that at least, we have a chance of getting one, scoring one, if not more, so that, we have one, vaccines that work, and two, enough production to meet the world's demand. Right? So I think that is hopefully a useful overview of the science that Ashley has outlined and the clinical translation that I provided, and happy to deal with questions later. Back to you, John.
Okay, thank you, Eng Yong. A lot of questions have come in and are still coming in, and I'll just ask you and Ashley to consider these before we come back to the Q&A session later. There's an issue of access. What is Singapore's priority to these vaccines later on? There are issues also and concerns about the mutation and the various strains of SARS-CoV-2 that are already there. How will that affect vaccine development? And the other issue of length of immunity, is that a significant issue? And reinfection. So these are some questions that have been coming through, and perhaps you can address them later. But now what I'd like to do is pass the time over to Amgad to give us an industry perspective on development of therapeutics and vaccines in the context of COVID-19. So Amgad, over to you.
Yeah, John, can you hear me?
Yes, we can hear you.
Okay, perfect. Okay, thank you very much. So
we can't see you though. We can't see you, Amgad.
Oh, well, I'm just trying to open. I try to start the video. Can you see me?
No, we can't. But anyway, why don't you carry on talking first and we'll fix this. Yeah.
Okay, I think because it appears that the host need to disable it. Okay, let me start maybe, okay, until we go down. So actually I want to give in the coming 5 minutes, maybe, covering the industry point of view in 3 talking points. The first one regarding the industry scientific heritage in developing new medicine. The second one is the partnership from the industry point of view with the government and academia and all the societies. And the third point, how is the industry advancing the battle to try to get like more new vaccine and new medicine. So I think the first one, we are all in the industry now trying to put all our scientific heritage in order and our expertise to try to find any molecules or vaccine. And just to give you an example from Pfizer point of view, we have been over the 170 years developing hundreds of molecules, and we're trying to put all our expertise in all the therapeutic areas to try to find this battle. And I think the vaccine, which is now everybody is also looking for that. Pfizer, in the last 138 years, has been developing a lot of breakthrough. I'm not going to go on that, but I just want also to give example of how is industry also partner with the governments that in 2 examples of vaccine. The first one, the smallpox vaccine, which was developed in the 1880s, and that was a disease killing also millions of people across all the world. And the second one is the oral polio vaccine. So that's also one of our industry and Pfizer also commitment. The second, I think, important point is how is the industry is aligning all the plan in order to accelerate molecules and vaccine manufacture and supply. And for us in Pfizer, we have identified like 5 points which has been mentioned clearly by our CEO, Albert Bourla, in the public. And the first one is we are sharing all the data of the scientific update of all our molecules. And we are not only sharing with the industry, but we also sharing them with all the government, all the academia, because I think this is the time where everybody is aligned together. The second part also of the plan, we are putting a complete expert of people from epidemiologists, infectious disease specialists, virologists, and epidemiologists. And a lot of these people in Pfizer are coming from the academia, university, and the CDC. And we are trying to put together how we can accelerate the development. The third important point, which I think some of the question is what we are doing in terms of registration. And in Pfizer, we are working as part of the industry with a lot with the FDA and our clinical research in order also to find an accelerated registration path in case we could find a positive molecules, whether therapeutic or vaccine, so it come quickly to the public. The fourth important point is how is the industry is dealing with the manufacturing and supply. And I think this is also very important. And I just want to assure also that, for example, in Pfizer perspective, we have ensured that we put additional funds for accelerating our manufacturing, facility for early production in case we could find any positive molecule or vaccine. So we will ensure that we will move quickly with all of the, the funds for the manufacturing point of view, and also the supply that we can produce millions of doses in a short period of time, so we can go to the globe in a very short time. The last important point in this plan is our alignment with the, with the regulatory bodies, with the FDA, with the CDC, with the NIH, in order also to put the research plan and work together in order to accelerate the time of the clinical research and the outcome. And the last point I just wanted to share, because there's a lot of question coming to us, about what the industry is doing, what is the type of the therapeutic and vaccine. And from Pfizer perspective, we identify 5 plans of molecules and vaccine working on. So we are working on one of the antiviral, compound, which showed some activity on the SARS-CoV-2 vaccine in the preclinical study. In terms of vaccination, we aligned also agreement with the German BioNTech company to develop a messenger RNA vaccine, which we hope that it will come to clinical trial in phase 1 end of this month if we got the regulatory approval in the US and Europe. We are also providing an additional analysis and additional data and analysis for one of the ongoing products we have for erythromycin. And I just wanted to be sure that this product is not registered for viral infection, but we are trying to provide all what we have from additional analysis, and we publish in a peer review that in terms if this molecule can provide any benefit to the COVID-19. And one also of the important, the fourth aspect, we are working in the UK and in Liverpool with 2 important research that we are trying to find any link or correlation between COVID-19 and the secondary bacterial pneumonia caused by Streptococcus pneumonia, in an effort to see if the pneumococcal vaccination can provide any value to the COVID-19 population. And last is also we are looking for one of the current therapy, the JAK inhibitors, the tofacitinib, which is originally for rheumatoid arthritis, but we are trying to see if that molecule can provide any benefit in terms of pneumonia with COVID due to its action on interleukin-6. So maybe this is like just quickly our 3 top, points from industry point of view, and Pfizer we are trying to capture and work on COVID-19. And I would just revert back to you, John.
Thank you very much, Amgad. Perhaps a question you could come back to later is the degree of cooperation and collaboration across the companies, because I think that's of interest as well. Yeah. But now thank you very much to all the panelists for sharing your thoughts. I'd like to come back to try and deal with the questions. We've had this overwhelming flood of questions, so we're trying to focus on the key themes, and we will try and address them because some of it has already been covered by the presentations. But perhaps I can come back to Yi-Sin first. There was a question that was raised because you mentioned the quite low fatality rate that we've experienced, which is of course a good thing. Are there any thoughts on why this should be so? Is it linked to the level of medical care, the patient profile? Is there some genetic predisposition? So Yi-Sin, could you tackle that question first?
Well, I did mention that we should not be too complacent because we are seeing a total low case fatality rate. Basically, it's also because we are having now a group of very young foreign workers, and therefore you don't really see the kind of age impact of a disease severity outcome. Now, let me backtrack a little bit just to share with you some information that we have looking through our patients' records. It is very clear in terms of the disease severity, it is highly associated with age. In other words, the older you are, the higher the risk of getting a severe disease that require either oxygens or require ICU care. So the younger the populations that you have with COVID-19, the lower the incidence of severe disease. So there again, this poses a lot of medical challenge as well, in the sense that how do we risk stratify them as early as possible so that they can be cared for at the appropriate care site. So I think for many of the local residents or people that follow the evolutions of the entire epidemic at this point in time, you will realize that there are a lot of community isolation facilities that are being set up in Singapore, and those are the facilities that now we are putting in patients that are either recovered while waiting for viral clearance, or individuals with very low risk of disease progressions, and we're putting there to monitor them throughout the entire course of clinical illness. So I think, again, I'm going to say that try not to be complacent, because we're just having a different case mix, demographic case mix at this point in time. However, I still want to give credits to our intensivists and of all the ICUs in Singapore in doing a remarkable work in terms of giving patients the ICU support, and we have very good intensive care outcome in the local setting. Over to you.
Thank you very much, Yisin. And yes, I think we want to emphasize the great debt of gratitude we owe to all our healthcare colleagues and all the others who are working across the institutions and also dealing with the dormitory situation at this point in time. But before I leave you, Yisin, can you make some comments about other therapeutics like hydroxychloroquine, convalescent plasma? So although you mentioned there's nothing effective, there seems to be a great deal of interest about these sorts of potential therapies, and also repurposing existing things like BCG vaccination. Over to you.
Well, I would think that this is not totally unanticipated, because if you have brand new conditions and you do have a spectrum of cases, severe cases that succumb to the infections, people will try all means to look into therapeutic approach. And the commonest way of doing it is look at some of the cell-based study, looking at the inhibitory level. From there to select some potential available medications to repurpose it for the management of patients. That is not something unanticipated. However, I would say that at this point in time, many of the information is being pushed out through different channels, including the high-level journals. However, did not give a very good and convincing evidence that any of all these therapeutic approach at this point in time have shown to have any efficacious outcome. So that is the key issues. And coming back to hydroxychloroquine and chloroquine, I understand that there are a lot of reports coming out, many of them observational, many of them not well randomized, showing clinical efficacy of chloroquine, high dose, as well as high dose hydroxychloroquine. Now, I also can share with you that many of all these trial sites, in fact, have terminated the use of chloroquine as a therapeutic approach because of cardiotoxicity. So that brings home a key message that while we're experimenting all these unproven therapeutic options, we have to be very careful in terms of monitoring and not to cause harm. The reason why is because we also know that 80% of the patients will recover and they do not really need a lot of medical care or medical attentions. And what is more important is first to do no harm. And that is my advice to many of them who wants to look into various therapeutic options, to study very carefully and again understand that whatever we have right now is still experimental at best. Over to you, John.
Thanks, Yisin. One more set of questions for you. The issue of potential reinfection. We're seeing these reports coming from South Korea and China. What are your thoughts on this? And also the broader issue of herd immunity. What are your thoughts on this in terms of allowing this to develop and potentially managing the situation as we go ahead in the next few weeks and months?
I think the term being utilized right now in terms of reinfections, it is really misleading because we really do not have good clinical information and evidence to suggest that this indeed is because of another episode of infections. So we are looking at these situations with a few hypotheses at this point in time. One is that we know very well that the virus sheddings can take place, and it can be a very prolonged virus shedding and sometimes intermittent virus shedding. And therefore, having 2 negative PCR and a subsequent positive PCR cannot by itself equate to a new infection. The second thing is that for any of the acute viral respiratory infections, it is anticipated that some of the respiratory symptoms may persist for a prolonged period of time and sometimes may appear to be worsened. Right? For example, cough. Cough tends to be the last symptoms to go away, and sometimes the patients may feel exacerbations of cough after they're being discharged. But when we re-examine the patients in terms of X-ray and in terms of looking at potential other organs involvement, as well as biomarkers, we have no evidence that indeed that could be episodes of reinfections. And of course, PCR can be very sensitive, and it flip-flop. It depends on the level of detections. So it can be very low level of detections that's been negative, and somehow it can be so sensitively picked up as a positive results. So those are a few hypotheses that we put in place. However, I still say that this is a very new disease, and we are still monitoring the situation very closely. We will not close the book. We will continue to monitor this situation very closely till one day we can really very convincingly, undoubtedly be able to prove that reinfections does exist. Herd immunity, I think, is a very loose term that people use, and it is a well-known phenomenon for many of the viral conditions that once you reach a certain level of immunity in the community, you have the ability to retard, slow the transmissions or stop the transmissions. Because basically you don't have any more susceptible individuals for the infected persons to infect. I think we have to monitor this situation very closely in terms of how COVID-19 will behave. And there are a lot more studies. I think Ashley as well as Leng Fa and Ing Yong's group studying together with us looking at the post-recovery immunity. I don't think at this point in time all the evidence are that clear in terms of the immune protections, the longevity of the immune protections and things like that. As a clinician, I think I have a very open mind. I would like to continue to study this in order for us to understand more. Over to you, John.
Thank you very much, Yisin. So now moving back to Ashley and Eng Yong and continuing this discussion on immunity, when we're thinking of vaccine development, what are the kind of problems that will be faced here, especially when the levels of antibodies in the population will vary? How does it affect clinical trials? And can you make some comment also on the observation that coronavirus itself has faced some difficulties with vaccine production in the past? And I think Ashley had touched on this earlier in relation to antibody-dependent enhancement. Would you say a bit about this? So who shall we start with? I think I'll invite Ashley to say a few words first, and then Eng Yong. Over to you, Ashley.
Okay, so I can maybe explain the phenomenon of antibody-dependent enhancement in a little more detail, and then I'll hand it to Eng Yong for more of the clinical applications. So antibody-dependent enhancement is the idea that when a virus is floating around in your body, it can bind to antibodies, and those antibodies should neutralize the virus. But in some cases, if they're weak antibodies or they're not effective at neutralizing, the antibody itself is supposed to be sort of a hook for other cells of the immune system to be able to what we call opsonize, so pull the virus in and break it apart and start presenting it to other parts of the immune system. So when that happens, you can have a situation, if the virus is not killed, of facilitating infection of those cells that have the receptor for the antibody. So this is something we want to make sure doesn't happen. It can theoretically lead to a higher antibody titers, or it can theoretically lead to increased production or release of immune mediators from immune cells that could potentially be harmful. There's some indication in this disease that inflammatory mediators are quite important for the pathology that occurs in the lung. So there's a fine balance of generating an immune response that's protective, but at the same time, not at all harmful or too exuberant to cause tissue damage. And so we have to make sure that that is a very fine line. And I think that's not only a question with the vaccine production, and it's just something that needs to be ruled out. It's not necessarily going to happen, but it's also a question about the convalescent serum trial. So we need to make sure that doesn't happen with convalescent serum as well. And there are a lot of factors that influence it in terms of antibody concentration and things along those lines. So this is something that will need to be monitored long-term in vaccinees as well. And with that, I'm going to let En-Yong speak a little more detail about the other aspects that you raised.
Thanks, Ashley. I totally agree with Ashley about the immune enhancement. I mean, she's outlined the process and all that. I'd like to, perhaps one more thing to add is that unlike for dengue, how this immune enhancement idea came about for dengue was it was first seen in patients where the doctors noticed that they were having the second infection, and that seems to be worse than the first infection. Where so the evidence was primarily clinical to begin with, and then came the lab workup to show the processes and all that. For coronavirus, except for cat coronavirus, for human coronavirus and this coronavirus too, it's the other way around, right? We have the lab evidence, but actually there's not a clear clinical support for this. So I think that's where we need to take this idea that there's the immune, the antibodies and all that could be harmful. We have to take that with a pinch of salt right now, and it's something for us to study. Then the question of, okay, it's been difficult to develop coronavirus vaccines in the past. That's true. But having said that, we didn't have the technology that we have today. For instance, for SARS coronavirus, the original 2003 outbreak, everyone focused on using a killed virus as a vaccine. And that was the only thing we had. Today, we have a lot more technologies, right? We can produce subunit vaccine, we can use DNA vaccine or RNA vaccine. And these are actually faster to make than trying to grow the virus, which is, you require the BSL-3 level of containment, and then trying to kill the virus. And if you don't kill it properly, then it's not safe. The other thing about killing a virus is that we assume that the virus will preserve all its features when it's in a killed state, because we use a chemical to kill the virus. It turns out not to be the case, and it's happened many times. The original killed measles vaccine made measles worse. The original killed respiratory syncytial virus or RSV vaccine made RSV worse, right? The killing process distorts the image of the virus, and so our immune response our immune system then sees a very different picture to what it would see if this was a live infection. So I think we are fortunate, I guess, today that we have other ways of making vaccines, and we can make them in ways that perhaps mimic the virus more accurately. And certainly the RNA vaccine and DNA vaccine, the part of the virus that we want the immune system to recognize is made inside the body, just as how the virus would make it. So then we think that will be a more accurate representation of the virus. I'll leave it as that. Anything else you want us to touch on, John?
Yes, En-Yong, could you say a bit more, since you're talking about messenger RNA vaccine, can you say a bit about the Arcturus project, whatever you can say, and maybe give us a sense of which organizations in Singapore are working on vaccine development and what they're doing? Over to you.
Yeah, so thanks, John. And in Singapore itself, we're working with partnering a company that's based in San Diego. Its name is Arcturus Therapeutics. Why we chose to work with this company is, again, it's got a platform that develops an mRNA vaccine. So an mRNA is like it's the code, the message to make the spike protein or the crown of the virus, which is where the key that the virus uses to unlock the cells to get into the cell. So if your immune response can target this part, then you effectively disable the virus, right? So many other companies, as AMGAT referred to, are also developing mRNA vaccine. The one interesting feature with Arcturus's vaccine is that the mRNA replicates. So you can put in, say, 10 copies of mRNA into the body, and then it will make, each one will make 10 more. So you may get 100 copies at the end. there's several advantages to that. One is that you put in a lot less to begin with. Two, it lasts longer. And those two things are necessary. I mean, the second one, that the vaccine stays longer in the body, is very important because the immune system then has time to recognize the antigen or the part that you want of the virus that you want it to recognize. If you give it too short, then it's like, it's like if you were to drive past a crowded street and then I point out to you, oh, that's my friend, and you go, oh, okay, but you will probably not be able to recognize this person because it just flashed by. Whereas if you have time to keep looking at the picture, then you develop better memory. And with better memory, you respond more rapidly and effectively when you actually see the virus. So that's the reason why we chose to go with Arcturus Therapeutics. Where we are today is we're in animal studies. I can tell you it's looking quite good. And I can't say any more about that, but it's promising. And we hope to get to clinical trial in the next 2-3 months.
Okay, thank you, Eng Yong. One last question for you. What advantage is there to Singapore in being involved in these trials in terms of eventual access?
Thank you. Yes, I think there were questions raised about, will we have access to the vaccine? And that's partly why we worked on this with Arcturus, so that part of the deal was that after showing that this vaccine works in a clinical trial, Singapore has priority to the first 5.5 million doses of the vaccine, so that we'll have enough to vaccinate everyone and protect us from COVID-19. Now, if I can take that one step further, some people have asked, is vaccine a necessary thing, the only thing to fighting COVID-19? I don't think so. I think vaccine is a part of it. I think Yi-Sin already alluded to the fact that, our patients do well, but if we can even prevent those, small, say, 5% or so that may require oxygen, and the fewer that may require ICU, if we can prevent that entirely, then we can get back to normal much faster than if we were trying to, rely on getting a vaccine and then preventing everyone from getting COVID-19. I think everything helps. They all complement one another. One does not supersede the other. I think, I always think of this fight, overcoming outbreak as a football game. You don't go into a football game saying that I'm going to take one shot at goal and that bad shot better go into the goal, otherwise I will lose. Nobody plays a game like that. And so you try multiple strategies. You go at the goal at different angles, and you do all you can to win. And I think that's what we need. And I don't think we can. It's wise to put out all our eggs in one basket.
Okay, thank you, Eng Yong. Can I come back to you, Amgad, now in terms of talking about the industry perspective and what companies are doing, either in terms of collaboration to move the vaccine discovery development process forward? And could you say a bit about the pricing issues, especially when eventually it comes to access across more highly developed countries and low and middle income countries in time to come? Over to you.
Yeah, sure. Thank you, John. I think there are 3 points here for your question. The first one is the level of alignment between the industry, and I think this is the time where all the industry are working as a one team in terms of sharing the scientific information. But an important point in this pandemic is also the alignment between all the companies in the industry, between the big pharma and the small biotech. And I can share a number of examples. I shared earlier Pfizer, when we aligned with the BioNTech company. But if you see more of the 70 molecules in the WHO report, or even there are 20 molecules are starting the phase 1 within this year, the different phases. And most of these molecules with a different platform and technology are alignment between small biotech company and a bigger pharma, bigger pharmaceutical. You probably also read about even companies like JSK and Sanofi that are partnering together. So this is the time where I think it's important to work as a one team. And I think the example of the penicillin production few years before, during the Second World War, were example of that, where most of the companies produced the penicillin where it was not, the one they are discovered. I think the other important point also, the alignment also at the same time with the regulatory bodies like FDA, like other regulatory bodies, in order to ensure that we go in the design of the clinical trial in a little bit more adaptive way, that, you know, we would just wanted to also to have, you know, the 10 years in just in 1 or 2 years. And just another example of the alignment, we are also working together as part of the industry to ensure the manufacturing and supply, there will be no issue. And probably one also of the additional advantages, for example, of some of this new technology like the mRNA or the DNA, that you produce a small part of the vaccine. So one of the advantages, if we can identify the good candidate vaccine, we can produce millions of doses in a very short time. And one of our commitment as an industry, that we will ensure a huge amount in a very short time, and we will work on access to reach different parts of the world. And I just wanted to let you know that, for example, we even asked our headquarters in New York if we can include in the COVID-19 vaccine Singapore as one of the centers, you know, because we know that we're going to start phase 1, although that some of the companies like Moderna, they started in the last few weeks. The first one, Pfizer and BioNTech, we are going to start the phase 1 end of this month, provided that we get regulatory approval. But there are 20 molecules that can start, you know, the phase 1 within this year. I think the other important point also, John, you mentioned, is how is about the pricing strategy. And one of our commitment as an industry at this point, that we are looking for the production of the vaccine regardless of the cost effectiveness model. And I think from Pfizer perspective, our CEO has announced that when we develop and we put our resources in the manufacturing for the COVID-19 vaccine, we didn't look at that point to the cost effectiveness model or the return of investment. We ensured that we put all the additional funds for the research, for the manufacturing, regardless to that point. I think this is a commitment for most of the industry at this point. And this is the reason you see that some of this alignment with the biotech happened even before having a full, complete deal, because I think the time here is important to the whole community, you know, to work together. So maybe just, I wanted to answer this very clearly because there were some, you know, questions about this point.
Thank you very much, Amgad. I think also to complement what you said, I think for the audience's knowledge, the other important players are the non-governmental organizations in vaccine development. So one particularly important one is CEPI, the Coalition for Epidemic Preparedness Innovations, which has significant funding from the Gates Foundation and the Wellcome Trust. And they are committed to raising about $2 billion to target 8 vaccine candidates. The other thing, a question was raised, or some questions were raised, how do you really expedite this 15-year normal vaccine development to what we're hoping is only a 12 to 18-month runway? And a lot of it means things are going on in parallel. As Amgad said, there's rolling submissions, adaptive licensing. But people like Bill Gates, they're investing a huge amount of money, and I think the industry too, in getting up manufacturing facilities even before you know whether a vaccine candidate will be successful. So it's a huge investment, but it's necessary if you want to expedite once you've tested for safety, efficacy, and then you really ramp up production to reach that kind of level. So it's a completely different ball game in terms of the way vaccine development is going on in an emergency situation. Now, time is catching up on us, and I would like to come back to each of the panelists in turn, starting with Yi Sin. What would be one takeaway that you have for us this afternoon? And also, what are your thoughts? Because many questions have been raised on this. How do we get back to normal out of this circuit breaker mode as soon as possible, but without further clusters surfacing? So one key takeaway from the points you shared this afternoon, and how do we get back to normal? So can I start with Yi Sin and then move through the panel and come back to Amgad? So Yi Sin, over to you, please.
Well, this is a very common question, but I think it's the hardest to answer. How do we get back? How do we exit successfully? And how do we get back slowly back to normal? I don't think the process is going to be a quick and easy one. I think it's going to take time for us to adjust along the way in terms of how we will get back into a normalcy. But I would say that, you know, what we experienced in the past as normal in the future may not be the same So we just have to keep a very open mind in terms of how this virus will force us to change many of our human behavior. Over to you, John.
Thank you, Yisin. Ashley, your final thoughts, please.
Sure. So I would say maybe my final thought would be the importance of scientific rigor in approaching these questions, because I think there have been a lot of studies coming out with, lacking control groups, for example, and we really need some basic research still as well. We need some research to understand what kind of immune response is protective. There's specialized responses involving the mucosal tract, for example, mucosal immunity that may be required. And we need to do just basic research to also understand how this virus is infecting and if it's doing something a little different than what we're expecting. A lot of what we are basing all of our decisions on is still based on the literature related to other viruses to a large extent, and to SARS and MERS and the studies that have been done in those systems. So I think we need some basic studies, and we also need to make sure when we move to clinical studies that they're very well designed and controlled, and it's worth it to go a little tiny bit slower if it allows us to have a decision that is very rigorous and based on evidence that is solid.
Thanks, Ashley. Eng-Yong, your final thoughts?
So I think, although we are working towards a vaccine, trying to find drugs and all that work, I think at the start of this webinar, Yisin has really pointed to the fact that our mortality rate is low. And I want to say that I think our mortality rate is low in addition to what Yisin has said, is that the number of severe cases have not overwhelmed our healthcare system. And to a large extent, our epidemic curve has been fairly flat until the recent spike, but fortunately it's in fairly healthy individuals at baseline. I think so my thought is that whilst we put in all this effort to develop vaccines and drugs, don't forget that simple, basic hand hygiene and sensibility can prevent infection. So please keep well.
Thank you. Very good advice for everyone. And Amgad, your final thoughts.
Yeah, I think my final thoughts, we would just need to continue as an industry, align together, but also aligning with the governmental bodies and the research center to ensure while we are developing, the adaptive way of the design, that we also looking not only for the efficacy of the vaccine, but also the safety. And I think the important other point is to keep also this, alignment in terms also of manufacturing and supply, and registration purposes, that when we also developing new therapeutics or vaccine, that, we put in place, that this is important to reach as quickly as possible to most part of the world. So we continue working on our manufacturing and supply at the same time of looking to the clinical design and, the vaccination and therapeutics.
Thank you very much, Amgad. I'd like to thank all the panelists, Yisin, Ashley, Eng-Yong, and Amgad, for your thoughts today. Unfortunately, I don't think we've been able to answer all your questions. Just in the Q&A alone, I see 144 questions. We've attempted to address the key themes as well as the earlier ones. I think this word unprecedented is something we keep hearing. I mean, this pandemic has really turned our lives upside down, but I think it's incredible the amount of collaboration, cooperation that the medical, healthcare, research, scientific community is pouring in to address this. And of course, as Eng-Yong's point was, everyone has a part to play in terms of simple hand hygiene, safe distancing, and so on. And so I think this is really, in a sense, the beginning of a longer journey. There are many things we've discovered, but so much more to discover. So I'd like to thank all the participants for joining us this afternoon. We hope you've learned something and it has been enlightening to a reasonable degree. And I would now like to pass the time back to Mrna to close the session. Over to you, Mrna.
I just want to take this opportunity to say, to echo your sentiment, thank you for all the participants to taking their time to share with us, and special thanks to all the panelists and to our moderator, John, of, having an excellent session. Hope that, as you said, people learn something and we can answer all the many questions slowly over time. I want to also use this as a reminder that we have another webinar coming up next Monday at the same time, which will focus on the business impact of COVID-19. And then our last last and final webinar session will be actually also next week on Wednesday evening at 6 p.m. Singapore time. That's because we have some international speakers based in UK and US, and so have to kind of have their have it at a reasonable time for them. So we look forward to seeing all of you again, and thank you again, panelists and moderator and all the organizers.
Thank you.